Hello everyone, welcome to our DZL webinar. I am Marco Lampe and together with my colleague Alexander Halavati, we will present you Introduction to Confocal Fluorescence Microscopy. As you all know, in biology we are bridging different scales from the human being in the meters range via tissues, single cells in the range of micrometers, down to organelles or individual protein complexes, down to molecules and even atoms in the angstrom range. Light microscopy is allowing us to bridge the scales unto a certain limit, the Abel diffraction limit. For resolution beyond that limit, we will need electron microscopy. In this technology, we will need to fix the samples and bring them into the high vacuum of the electron microscope, so we only can study fixed non-living specimen. Whereas in light microscopy, we have the advantage to also study dynamic processes in living cells, tissues, and even animals. The big advantage was the discovery of fluorescence microscopy. As you can see here in this phase contrast transmitted light image, we can see cells and even identify perhaps individual organelles, but we don't know their identity. In tissue sections that are stained with specific stains, we can identify classes of proteins, lipids, and, for example, mucus. But still, we cannot pinpoint an individual molecule. And this enables us fluorescence microscopy. Here you can see actin, the protein actin, labeled with the green fluorescent protein, GFP, in a cell plated on a glass cover slip. And you can see the individual actin fibers here formed by this protein by fluorescence microscopy. So we can really now see individual molecules in the cell, where they are located and how they interact with each other. Just to remind you, on fluorescent microscopy, here's a simplified Jablonski diagram. So you have the fluorophore usually residing in the ground state. Upon interaction with a photon from the excitation light, the molecule is excited, so an electron is catapulted to the first excited state. After a short time period, this electron is returning to the ground state and de-exciting the fluorophore upon emission of another photon, which is of low energy and therefore red-shifted. And with this process, we can really pinpoint and detect these individual molecules that we are interested in. But how can we label them in the cell? Therefore, we can make use, for example, of fluorescent proteins. We have a number of variants of fluorescent proteins. The most common one and most known one is the green fluorescent protein derived from jellyfish. But there are now variants from other species as well in different colors, ranging from the blue fluorescent cyan range to the infrared range. So we can really do multicolor imaging now with these fluorescent proteins, and we can genetically fuse these fluorescent proteins, which are stable entities, to a protein of interest and combine these or link these two with a flexible linker. So this allows these two proteins to move a little bit in respect to each other and minimize the effect of the 24 kilodalton of the GFP onto our protein of interest. Usually these proteins behave well in cells and close to the natural conditions, but of course controls are needed to ensure that the protein of interest is not hampered by this fluorescent tag. Another very powerful method developed in the 1980s is immunofluorescence. Here we're using antibodies to specifically pinpoint our protein of interest. As you know, an antibody is recognizing a small epitope, a small entity on a molecule. So you can even pinpoint on a protein certain parts of it just using the specific antibody recognizing this entity. But how do we do the staining for microscopy, for fluorescent microscopy on a cell? So let's assume here this is our cell on a glass cover slip, and this is our protein of interest. We would then chemically fix the cell to stop all process in the cell and be able to treat the cell with detergent to permeabilize this. Therefore, we make holes in the plasma membrane and in other interior membranes, allowing the antibody, which has a size of 10 to 15 nanometers, to penetrate through the cell and find the epitope and bind to it. Usually this first antibody, the primary antibody, is not fluorescently labeled. Therefore, for the fluorescent labeling, we are adding a secondary, so-called secondary antibody, which carries a number of fluorophores, usually 7. Since the constant part of the primary antibody can be recognized by many other secondary antibodies from a polyclonal serum, This is an amplification step, so we can enhance our signal. Furthermore, this is pretty practical because we then don't need to label each primary antibody individually with our fluorophore of interest, of choice, but we can just change to another secondary antibody, and we don't need to spend precious primary antibody to be wasted during the labeling procedure, which is always not 100% effective. So now we have the stained cells, and now we have to move on to the microscope. Our microscope, unfortunately, is far from being perfect. So whatever happens in a microscope is basically that we don't see the real structure. We are always seeing a blurred structure. So in the image, we're seeing a blurred structure. So assuming that this is a point source, so infinitesimally small entity, it will always appear larger and blurred in the microscope. And this is called the convolution process of the microscope. And this is also limiting its resolution. So we can't see in a small entity here. It always is blurred to a specific size, which is also the resolution of the microscope. In larger structures that are consisting of many proteins, and that are also labeled with many fluorophores, we can now treat each fluorophore as one of these point sources. And each fluorophore can be deconvolved individually, which then at the end, if you have a dense structure like here, these schematic microtubules, we end up with a pretty much blurred structure in the microscope. This, of course, can also be described in a formula. So we have our object, and we then have the convolution process with the point spread function of the microscope. The point spread function is correlated to the resolution of the microscope and then is giving our final image. So how is resolution defined? In the 19th century, Ernst Abbe developed this formula here that still holds true for any diffraction limited microscope. The resolution of the microscope, D, depends on the wavelengths of the detected light, or of the fluorescent light in the case of fluorescence microscopy, divided by 2 times the numerical aperture of the objective. This employs us a strict limit, which is in practical terms on a light microscope in the range of 200 to 250 nanometers lateral resolution that is maximum achievable.
To understand better the resolution of fluorescent microscope, let's have a closer look into the process of image formation. The resulting image, which represents multiple objects, can be seen as superposition of multiple images, each represent one single object. Let's start from a small object which will be located in a focal plane. When the light goes through the objective, in the image it will be represented by the focused nice dot. However, at the same time, objects which are out of the focal plane might exist in the same specimen. These objects will result in the image plane in a scattered light, which will add the haze to our image. We can have, of course, much more objects which lie below or above the focal plane. At the end, the resulting image will be composed of the objects which are in a focal plane and which are nicely focused, and the haze which comes from all other objects. This leads to the reduced image contrast and, as a result, to the reduced resolution and insufficient information from our image. This is the challenge which is in general addressed by confocal microscopy. Let's think what we demand from that first. As we've seen, we have a problem of out of focus light, and this will be in general the problem when we image thicker samples in, for example, living tissues. It will be also a problem for multilayer cell culture and also dense subcellular structures, as just seen on the previous slide. So what we want to achieve is we want to have out of focus light invisible. So we want to apply optical tricks to largely reject out of focus light, and by that to improve resolution. Let's now have a look how it's achieved by fluorescence microscope. We will start from the same concept as we had before, with a single object in a focal plane and its representation in an image plane. The difference now that in front of our image we put a small aperture called pinhole, which allows to go through the focused light and to reject the majority of out of focus light. So now if our out of focus object emits the light, most of this light will be rejected by the pinhole, and we still have nice information on our image. As a result, we will have better resolution. So let's have a look on the resulting image. We have only dots which are in a focal plane. However, we will not see all the information which is out of focus plane. The drawback of that is the intensity of the image is largely reduced, so we will need to expose the sample to much higher light to get the same intensity on our sample. Let's have a look on the dependencies of the resolution in X, Y, and Z on the pinhole size. The resolution in X, Y will depend on the pinhole size only when we use very small pinholes, which is never used in practice. However, resolution in Z will gradually decrease when we decrease the pinhole, and that's major achievement of the confocal microscope. When we close the pinhole below 1 area unit, the intensity of the image will suddenly drop down, so we have to always find a compromise between size of the pinhole, improving our resolution, and getting enough photon to form nice image. Further on, we want to see how it's implemented in the hardware part. So what we will use in our confocal microscope typically is the laser light, which will be then exposed to the sample through the objective, as nicely seen on this picture. Then the fluorophores which are inside our sample will emit the photons which will be captured by the detector. As we already told, to reject out of focus light, we will put pinhole in front of the detector, but in this case, the detector can capture only single point from our sample. It means that our image in this case contains only single pixel. To form an image which contains multiple pixels, we have to scan with our laser beam through the sample, which is done with the scanning module. So the scanning module composed of multiple mirrors, which allows the laser beam to scan in X and Y direction. So it will look like that. The laser beam will continuously go through the field of view, scanning line by line, and covering the whole field of view. The resulting image of the object, which is schematically represented by the cloud, will be, of course, pixelated and will be represented by the pixels where the signal is positive above the background. Now we would like also to acquire images in multiple fluorescence channels in order to be able to distinguish different molecular species. This can be nicely implemented on the confocal microscope by sending emitted light, which passes the pinhole through the prism, or another more complicated optical system, which allows to separate photons with different wavelengths in the different directions. In this case, the different detectors can be placed in different spatial positions to capture these different photons, and by that recording signal simultaneously or quasi-simultaneously in multiple spectral channels. As a result, we can see that we can acquire the 3D stacks in multiple fluorescent channels by moving our sample relative to the objective and acquiring with multiple detectors, as presented in this movie. What we can see is that we can, in this sample, we see the different cells in different layers, and this information we will not be able to capture with standard epifluorescence microscopy. So on the right part of the slide, we now see the superposition of all our individual images, which shows us the dense cells in our sample, but we cannot distinguish which cell belongs to which layer. Whether from individual slices, which we just saw in the movie, we can easily capture this information. Let's summarize everything what we talked by now. The confocal laser scanning microscopes have certain advantages over standard epifluorescence microscopes because they allow us to reject out of focus light, allow us to perform optical sectioning, and can make flexible experimental design with multicolor approach. However, they have certain disadvantages which propose us further challenges to improve the technique. One of the disadvantages of this technique is the slow speed, because we have to scan point by point sequentially. Another disadvantage is an image noise. And this is due to the fact that we focus on each individual point only for a very short time, and we capture very small number of photons from each individual point. The next point is resolution. Of course, it's better than resolution of standard epifluorescence microscope, but we don't get any improvement in practical situations in lateral resolution. The only improvement what we get is only in axial direction. Last but not least, we have to mention that we try to image deep tissues or other deep specimens. We will also end up with the problem of light scattering. Let's now have a look in the second part of our webinar, how these challenges can be addressed with more advanced techniques. So we will start with the speed challenge. When we talk about intracellular structures or intracellular organelles, for example, we want to acquire superfast images because we want to capture these dynamics in live cells. Very often, the speed of standard confocal microscope will be not enough for that. For that purpose and for some others, we can use a similar principle, which implemented differently in terms of the hardware, which is called spinning disk confocal microscope. In this case, pinholes are placed in so-called Nipkow disk, which is rotating with a fast speed. We will excite several pinholes at the same time, and by that capture information from several pixels at the same time. Another advantage of this technique is that rather than having point detectors, we will use camera systems, which in general have higher sensitivity, and by that we can reduce the amount of light with which we expose to our sample.
So now we are switching gears and coming to two-photon excitation or two-photon microscopy. Just a short reminder, so far we've talked about one-photon excitation. Here you see again the simplified Jablonski diagram. You can see here that the molecule or the fluorophore in the ground state, upon interaction with the excitation photon, it's catapulted to the first excited state and then emits the fluorophore of a slightly lower energy a few nanoseconds afterwards. The light is redshifted in this case. We can also employ a little trick, and that means we take two photons that have half the energy of the S0, S1 transition and basically let them interact with our molecule at the same time in less than 10 to the power of minus 15 seconds. And if we do so, we also can excite the fluorophore. We have the energy transfer happening, and then we get a photon out, but this is blue shifted, so this has a higher energy. But the most important part is that this interaction has to happen within this short time frame, pretty much instantaneously. So we need really a high number of photons in that area, so a high photon flux. This has a number of implications, and I will show you the most important one here, and why this is the reason why two-photon microscopy is very much appreciated in imaging larger organisms. So here on the top, you see the single photon excitation of a fluorophore in a cuvette here, in the liquid in the cuvette, and you see that here is the focus in the center, but above and below the focus, we are also exciting the fluorophore solution because the energy is sufficient to do so. However, if we have this interaction of two photons in a short time frame, and here we have this two-photon excitation, this near-infrared excitation, we only can excite the fluorophores just in the center of this diffraction-limited spot, just in this focal spot of this lens. And therefore we have a very confined region where we can detect, where fluorescence is emitted. This has the advantage that we can omit the pinhole in our system, so we don't need to use the pinhole, and we can detect all the light that's coming back, and we know where it comes from because by excitation we have defined it. So we don't need, in comparison to single photon excitation, where we're always exciting above and below our plane of interest, we can basically remove the pinhole. This has the advantage that if light in our specimen is scattered, we can still detect it and reassign it to the correct origin. However, as I mentioned, we need this high photon flux, so we need very powerful lasers, and also the microscope is increased in its complexity, and therefore this is not a standard technique, but usually used for live cell imaging or deep tissue imaging. And I will just show you now one example here. This is a Drosophila embryo, GFP labeled, and we will now start going from the bottom up to the top of the embryo, and you can see here labeled the step in micrometers. On the left-hand side, you see the confocal image. On the right-hand side, the two-photon image of the same specimen. And you can see after a while that in the center of the specimen, there is less detail shown in the confocal image, and at a certain time point, we are not even able to detect structure there, whereas in the two-photon image, we still can remain most of the structural information. So therefore, for a thick specimen and living specimen, even in vivo imaging in mice, two-photon microscopy is highly beneficial. Let's come to another method of improving image quality. Perhaps you remember the first slide on convolution. And if you remember that we are convolving the image with a point spread function, so we are blurring our image. Of course, you've seen the formula, and you might have asked already at that time, since we don't know the object, but we have recorded the image and we know the point spread function, how can we revert this process? Is that possible? And yes, it's partially possible by doing so-called deconvolution methods, sometimes also referred to as image reconstruction. And this method tries to restore the original image. However, this procedure is hampered by the fact that our image contains, for example, noise, shot noise, detector noise, and that also if we have a very dense sample, the solution of this procedure is not always unambiguously. Therefore, we can just try to estimate from our blurred structure the real structure. And therefore, we can also enhance contrast, enhance resolution, but just to a certain limit. And since this method is an iterative method that's trying to guess the best solution for the problem, we also have to be aware that artifacts can occur and therefore need some experience if you're handling this method. But if you're doing it right, you can achieve quite dramatic enhancements of your image quality. And I've now picked just one example here, one of species that doesn't have so much to do with medical approaches, but I personally like them. These are diatoms. They are living in salt water, for example, very beautiful marine species. And here we just stained the silica skeleton of these diatoms. And here you can see the confocal image, and you see here this area is pretty much blurred, whereas after deconvolution we can see this little tiny skeletal structure here much better resolved in the case of the deconvolved confocal image. Therefore, it's always good to consider deconvolution in your process of image processing and analysis. Next, I want to introduce to you some advanced techniques, also called super resolution microscopy. In these techniques, for example named rescan or rescan, we are employing the fact that after the pinhole we still have some information that we are losing if we are just detecting the light intensity as in a standard confocal microscope. By subsampling these information, just by putting the light on a small array detector, we can improve the resolution of the technology by 1.4-fold. In the so-called nanoscopy techniques like STAT and single molecule localization microscopy, also known as STORM, we can have an improvement of 5 to 10 times of the resolution of the image, which results in a resolution that can be below 20 nanometers in the lateral dimensions. I will not go into detail, but I just will show you the power of these techniques, and we will cover them certainly in some of the future webinars. So one example is STAT microscopy, and here you can see a 2D STAT image of a centriole. This is a tiny part, and you can see here this is 500 nanometers inside the cell, and we've labeled here with fluorescent microscopy 2 proteins, CEP152 and centrine 3, and in the confocal image you can see that they're slightly displaced, but only in the STAT image with a resolution of less than 50 nanometers we can really see how they are localized in space, and also see that the CEP152 has a 9-fold symmetry. This is STAT microscopy. I will now show you just another image of D-storm microscopy. This is the Widefield image. So this is what you would see if you look at the structure just with a camera. Now we are switching to the super resolution mode, and you can already see that we have here kind of the barrel of the CEP152 and the centrine located in the center. I will now show you an animation of this. Again, the CEP152 here in cyan, the centrine in magenta, and here on the right hand side the overlay. And please note that this scale bar here is 200 nanometers, so our structure is just a little bit larger than the 200 nanometers, just a little bit larger than the diffraction limit of the microscope. However, with this technique that has a resolution of less than 25 nanometers, we can see the fine structure in all 3 dimensions. You can also see here now the power of fluorescence microscopy. You see nicely the barrel of the SEP152 labeled, and also the centrin 2, and you can now see how they reside in this complex. However, since we have not labeled any other protein of this complex, we are just blind for them. We can't see them. So this is, on the one hand, the power of fluorescence microscopy, with, for example, antibody labeling, that you specifically see your sample or the protein of interest. On the other hand, you're just blind for all the other proteins that you haven't labeled. With this, I would like to end, and thank you for your attention to listening to this webinar, and hope to see you again in one of the next episodes.